Morning, everybody. So this morning I'd like to tell you a little bit about some of the results from our consortium, the DROPPS consortium. And so I am actually happy to report that we are the smallest of the consortia. That's certainly made my job, I think, a little bit easier. We consist of 6 institutions, all listed there. We have a relatively small number of PIs, research scientists, postdocs, compared to the list you've seen from some of the other consortia. We also do have a large number of both graduate and undergraduate students that have been involved with our project. I think, to me, the most interesting part of all this has been the fact that we have a mixture of engineers, biologists, and chemists. So I think we've gotten some really good synergistic results from that. I know it's certainly been a joy for me to get to work with some of these engineers and get to be able to apply some of the high-tech tools that they have to some of the biological problems that I'm interested in. So the DROPPS consortium, our overarching research goals really were just to try to understand, so we do more of sort of experimental approach. We want to understand the physical and chemical factors that affect how oil breaks up in the sea. We also want to then understand how that dispersed oil changes both chemically and biologically by interacting with microbial with the marine microbial bacteria in the sea, and also with the base of the food web with plankton, marine phytoplankton, and zooplankton. So we really focus on small-scale processes and then try to link these to larger scales with some mesocosm experiments as well as some modeling efforts. So again, I think the overall goal of our consortium was really to sort of have improved both empirical and theoretical understanding of how oil basically breaks up in the sea and how that's affected by these physical, chemical, and biological interactions in the environment. And so basically what we want to do with that is then be able to provide information, additional information that can be used for predictive models that will allow for management decisions about the risks and benefits of extensive dispersant application in response to marine oil spills. And our approach then is we've built within our consortium a number of specialized facilities to directly observe the breakup of oil under physical forcing. And in particular, we've applied some advanced optical methods, including high-speed video, holography, PIV, as well as using things like microfluidic devices to be able to study these, again, at sort of really micro scales or very small scales. We've also been very interested in sort of looking at both the lethal and sublethal effects of oil and dispersants on planktonic organisms at the base of the food web, and then also using mesoscale facilities to look at the biological and physical interactions between oil and plankton on larger scales with more complex groupings. And then finally, all this feeds into a modeling effort, again, to try to improve our ability to predict what happens to oil when it's spilled, and particularly when dispersants are applied. So this first slide shows an example of one of these specialized facilities. There are a number of them. I won't be able to go over all of them. This is a wave generating facility at Johns Hopkins University at the laboratory of Dr. Joe Katz. So you can see there's a wave generator over on the right-hand side, and sort of in the center top there's a component that then produces a fixed size oil patch on the surface of the water. And so next I will show you a high-speed video of a wave interacting with that. And so the top is oil without dispersant, the bottom is oil with dispersant, so you can see that oil patch was just released. This was taken at, I think, 250 frames per second and played back at 15 frames per second, so slow motion. But you'll notice on the top, the oil droplets are much larger compared to the oil droplets on the bottom. And so using various optical approaches, we can get a very good empirical idea of what the size distribution of these oil droplets is from this sort of, and again, this is just a single pass of a wave. Now, one other thing that they've discovered in the CAS lab, I think that is really interesting, is not only are these oil droplets being broken up and spread into the water, but they're also creating oily aerosols, sending some of this oil and water up into the air. And so in this video, you'll see in the bottom we've got the wave again. That little red square shows the area that's magnified on the top. And on the top, it's a series of 10 consecutive holograms recorded at 250 frames per second, and basically showing you these tiny aerosol drops that are then spread up into the atmosphere as a result of this breaking wave. And the data on the left-hand side shows the distribution, size distribution of the droplets that are created by the waves. So again, with no oil, these are just water droplets. As you add oil, and then particularly oil with dispersant, you can see more and more of the smaller droplets are created. So some of these are extremely tiny droplets and could be carried very long distances throughout the atmosphere. So another phenomena that had not really been considered before, again, this is from Dr. Katz's lab, is looking at how raindrops falling on the surface would affect oil on the surface of the water. And again, I'll show you high-speed video. So again, this is no dispersant on the left and with dispersant on the right. And you can see not only are smaller droplets created in the water, but again, you can see the aerosol droplets that are then falling back to the water surface. So again, this allows us to look at how this oil is again dispersed just from, in this case, a single rain droplet falling to the surface. And here's just an example of some of the data. So again, actually able to measure the size range of these droplets. And again, comparing at the bottom the size distribution under controlled conditions without oil. Again, just the water droplets in that crown that's created as the splash occurs. And then with oil, you can see again, there are many more smaller droplets created. So this is some work, again, really sort of focusing on the really small scales. This is from Jan Sheng's lab at Texas Tech. So he's devised some really sophisticated approaches to be able to actually look at the behavior of individual oil hydrocarbon consuming bacteria and how they interact with these really small oil droplets. So using this facility, he can look at oil with and without dispersants. He can actually enumerate the number of bacteria there, can look at their growth rates and so on. And for example, in this particular experiment, which he used cultured oil-degrading bacteria, it seems that the small size of the dispersed oil droplets, again, does not promote higher biodegradation rates. So even though smaller droplets might make the dissolved substances come out faster, it doesn't seem to promote biodegradation. And in fact, we're very much interested as well in, there may be a minimum size of oil droplets that can actually be colonized by hydrocarbon degrading bacteria, because some of these droplets are actually in the same size range as the bacteria themselves. And this is just another example of some of Dr. Sheng's work. So he's using, well, anyway, I guess I won't be able to start these videos. But anyway, he's using microfluidics in this case. So you can see in the lower left, here's the microfluidics device. He can make a single drop of crude oil on the end of that little projection, and then you can make a flow of seawater with hydrocarbon degrading bacteria flowing past that. So that simulates an oil droplet rising through the water column, so we can actually look at the encounter rate of hydrocarbon degrading bacteria with that. And basically what these results show, that when you compare it to bacteria-sized inert particles, of course they typically just flow around that oil droplet. With the actively swimming hydrocarbon degrading bacteria, there is a higher encounter rate. So then, of course, there are a number of mechanisms that can be used for these hydrocarbon degrading bacteria to break down oil. A number of the processes occur best in anaerobic conditions. So often these bacteria form biofilms on the surfaces of these oil droplets. So Kate Stebbe at the University of Pennsylvania has been involved with looking at the biofilms of these bacteria on oil-water interfaces. So you can see here, so initially these bacteria, when they settle on the surface and first form these biofilms, they're still active. You can see they're moving around. Then eventually this film becomes more viscoelastic, and then finally it becomes just more solid and elastic. And this, basically what this shows in this stage, so here's a robust biofilm formed on the surface of an oil droplet. And you can see from this little video clip that it's strong enough so that when you withdraw the oil from it, it retains its strength and actually gets wrinkled. So as a New England Patriots fan, I think this is the real inflate gate, or deflate gate here. But anyway, so you can see it really is going to change the characteristics of these oil droplets. So it's going to affect how they break up in turbulence in the sea and the rate at which dissolved compounds will come out and so on. So this is very important information as well. So in my lab, we look at how zooplankton are affected by these small oil droplets. We've looked at everything from marine protozoa through copepods and up to gelatinous zooplankton. We've done all sorts of different experiments. We've gone out on the pelican, for example, and collected natural assemblages of planktonic organisms out in the middle of the Gulf near the Deepwater Horizon site. We do shore-based experiments in Texas. We've also done laboratory experiments with cultured organisms and then expose them to oil and dispersants and look at both lethal and sublethal effects of oil and dispersants on them. So here's one of the interesting repercussions of dispersing these oil and creating these really small droplets is the fact that these then become in the size range of particles that can be fed on by zooplankton. And basically what we found is that zooplankton eat them without any problem whatsoever. So here's some examples of some work that shows that, for example, these heterotrophic dinoflagellates, so single-celled protozoa, will actually ingest these oil droplets. So for example, you can see in figure B, there's a light micrograph. You can see the dark food vacuoles within. To confirm that these are actually oil droplets, we can apply UV epifluorescence illumination, and you can see then that these fluoresce under that, so you can see which ones are the actual oil droplets. So again, we see actually very active consumption of oil droplets by heterotrophic dinoflagellates like Noctiluca. We found similar things in copepods. So for example, you can see here in a number of different copepod species. Again, the figures on the right-hand side show copepods on the left of those panels taken with light micrographs, and again, the other on the right-hand side with UV illumination. And you can see that there's actually small droplets of oil within the guts of these zooplankton. And I guess even further proof, of course, some of you may not recognize what the two objects on the lower left are, but those of you that work with zooplankton are certainly familiar. Those are copepod fecal pellets. Okay, and you can see the one on the right on the left rather, under light microscopy just looks like a normal fecal pellet, but you can see again on the one on the right of that under epifluorescence microscopy shows that it's packed full of small droplets of oil. So again, could be important because fecal pellets sink, and so actually by consuming these oil droplets and passing them through their guts, this may affect the balance of some of the oil in the environment. We've done lots and lots of different toxicity experiments, so I'm just going to show this one result, which is I think a good general representation. Although again, I would mention that every group is a little bit different. But basically, what we found as a sort of broad brush approach is that generally the crude oil itself is less toxic than even the dispersant itself. And again, that's in the appropriate ratio, so using 20 times as much oil as we do dispersants. Typically, just the application of the dispersant itself is more toxic to zooplankton than the oil. And in fact, dispersed oil is the most toxic mixture. So again, it varies a lot from group to group. For example, with protozoa, we find that dinoflagellates are much more resistant to the toxicity of oil than things like ciliates. Jean-Fei Liu, our chemist, has certainly been involved as well in looking at how the chemical composition of crude oil changes with both in the presence of hydrocarbon degrading bacteria as well as other environmental conditions. And so this is really important for us in terms of our toxicity experiments, looking at both lethal and sublethal effects, because we need to really consider this, that the fact that of course the oil is, as Nancy mentioned, is an extremely complex mixture, but it's also continuously changing. So if you're just looking at the effects of crude oil, it's not going to be really realistic because, again, the oil is changing as time goes on in the environment. Finally, we've also looked at a number of different, we have some specialized facilities that we can use to look at, again, oil under sort of a mesoscale situation. So one of our colleagues is the group at Syntef. They have that large tower basin at Syntef where you can inject oil into the bottom of the facility, and also you can inject dispersants, and so that again allows us to make empirical measurements of the size distribution of some of these oil droplets. The facility On the right is a tower system that we built at the University of Texas, and there's a corresponding one at Johns Hopkins University. This one's a little bit different. You can see it's narrower, but basically what we've designed here is a system, and you can see all the windows in it as well. So this is one where we can actually observe the interactions between rising oil droplets and planktonic organisms. So there's a number of different layers of windows there, but basically what we do is we inject small droplets of oil into the bottom and watch them rise, and we set up density discontinuity layers within that column so that we can set up thin layers of planktonic organisms. So far we've looked at some different phytoplankton species, and so we can actually look at the interactions between those rising droplets, oil droplets, and some of the different planktonic organisms. And again, finally, all of this is to really, the goal of this is to try to help improve the oil distribution model that SynTef has developed. I think they've been working on this model, I think they told me since 1995, so they're continually trying to improve it. They've added some components to account for sediment oil interactions. We've also working with them to include some components looking at the bacteria and plankton interactions that we've described so far. And again, all these things will hopefully allow us to better predict what happens to oil as it rises up through the water column. So how do we use these results? Again, I think the ultimate goal is to try to use these results to inform models, that allow us to look at the environmental, economic, and even human health impacts of oil reaching the surface, either where some can be recovered, but there's also a possibility that some may move into sensitive coastal environments, for example, if you didn't apply dispersants, versus what the impact would be on the environment if you did apply large amounts of dispersants. So again, we want to provide, I think, some more information that'll allow these decisions to be made as to whether it's more reasonable to apply dispersants the way they did in the Deepwater Horizon, or whether in some circumstances it might be better not to do that. In terms of the major gaps, I didn't think I'd have time to talk about this much, so I would say see our poster, but I'd say the one thing that we are adding is a human health component in our next round, and so we're very much interested in these small aerosol droplets that are created, and so we're interested in how this may affect human beings and wildlife as well that might inhale these droplets. Some of them are small enough so that they could actually be inhaled very easily and actually pass directly into the bloodstream. And so with that, this is a little figure from Rudy Strickler in our consortium, so you can watch a tethered Daphnia juggling some little oil droplets there.